What's up, everybody? We're doing membrane transport tonight. I wanted to go through as much detail as I can, so this one might be a little bit longer. Also, based on some of the requests, we're going to try to switch things up a little bit and, show you my face. I don't know what the deal is with that, but let's hook you up. And we'll do it right away on the first slide. You ready? All right, so here's Springfield up further north. Here is Enfield further south, and we got the mighty Connecticut River that flows down into the Long Island Sound. Definitely has a direction to the particles within that river. I hope everybody understands it would be really easy for us to jump in the river and flow down south. And for that example, we'll show you Mr. Wiley swimming. Here we go. Sun's out, guns out. All right, so if I get ready to jump in this river here, you notice what happens when I get into the river. I'm going to flow very easily without any expenditure or energy down that river, okay, with the gradient. And how this relates to what we're going to be discussing is that anytime we're talking about particle flow or movement, you have a gradient that is providing kinetic energy in a direction, okay? So when particles have the ability to move with that gradient in that same direction, it's a passive process. It doesn't require the input of energy. We don't have to alter the shape of any proteins to get that job done. It's just pretty much effortless if we have the correct conditions and the correct machinery to get that done. Okay, so let's look at a couple different examples of that. The first one being here where we have a phospholipid bilayer. It's our protein right here. Okay, so you notice there's 3 particles of this nonpolar oxygen gas on this side of the membrane. Over here we got a whole bunch of them. Okay, so in terms of movement of particles, these things are naturally going to want to go from their high concentration to an area where they have less, or to a low concentration. All right, so the first particle here is going to move across the membrane, and if you notice, that did not have anything to do with this protein. That particle went directly through the gaps in this phospholipid. Okay, and small nonpolar molecules like this, nitrogen gas, carbon dioxide, all of those things are going to have the capability to move in a very similar way. So these oxygens are going to keep coming across this membrane until we have the same number, or a similar number I should say to be more accurate, on both sides of this membrane. Now over time, if let's say we had 11 particles here and 10 particles here, you're always going to have movement of particles. That's why we use a term in science called dynamic equilibrium. It's a concept that we have equal numbers on both sides, but it's not constant. They don't stop moving at that point. They're going to continue to move, but they're always going to move in that direction from high to low. So if we were to pause life and take a picture of either side of the membrane, it's possible you would have 11 and 10 on one side, 11 on the other. But I hope you guys get the idea. These particles would continue to move, and in this case they're small enough and meet the criteria to be able to go directly through these phospholipids. We call that simple diffusion in biology, and a very important part of the maintenance within cells. Okay, so just to reiterate, simple diffusion is the movement from high to low concentration through the phospholipids. Again, really important that everybody can identify that this has nothing to do with a protein, and there is no energy expenditure in this process. All right, let's look at a second example here. This time we have actually a charged particle. Okay, so these are sodium ions. Again, low concentration on this side of the membrane, high on this side. Okay, and so what's going to happen in this particular case is that sodium is actually going to utilize this protein. So charged particles are not going to be able to fit in here because of the polarity and some of the characteristics of a membrane. Charged particles don't readily pass through the middle of this. So anytime we have a charged particle like this sodium, we're going to have to utilize a protein. And this again is a type of passive transport. It does not require energy, but because we're utilizing a protein to do this, the protein is helping. We call that in biology facilitated diffusion. So this is again simple diffusion, but it's happening with the help of a protein. So simple diffusion is through the membrane, and facilitated diffusion is through a protein. But the energy expenditure is exactly the same. There is none. And also it's down the gradient from high to low concentration. Okay, so facilitated diffusion, high to low concentration, without the expenditure of energy. And again, this part of the term refers to the utilization of a protein. Okay, so let's look at a third scenario. This time we have water and we have sugar. Okay, now sugar is very large relative to some of the particles we've been talking about, and over here we have a high concentration of sugar, and over here we have a low concentration of sugar. But in this environment, we also have water present, right? So over here we have 3 particles of water, over here we have 7. I've numbered them to try to keep it simple. So over here, just to reiterate, sugar is high, but water is low. And over here we have water is high, but sugar is low. So what's going to happen here is these sugars are going to want to move in this direction, from left to right. Okay, and they can't go through the protein, they can't get in between the spaces, so the sugars are in trouble here if they got to move. They're not going to be able to cross that membrane unless we have some kind of a transport protein or some kind of a way to open up a channel for these things to get through. The water, on the other hand, is going to be able to move. Okay, now if we go back and look at this again, you had a situation on this side of the membrane where 70% of it is water based on the number of particles and 30% sugar. We're just using these numbers to try to keep things simple. Over here was 30% water and 70% sugar. So we just showed again that sugar is not able to move, but water can. And water can move through the phospholipids in small quantities. So when we look at this, you just saw this particle move across. And now we have 1, 2, 3, 4 on this side. And over here we have one less, so we have 6 particles on this side. Okay, we have the movement of another particle in that direction. Okay, so now on this side we got 1, 2, 3, 4, 5. Over here we got 1, 2, 3, 4, 5. Okay, so when we look at the concentrations, what has happened is we've gotten a lot closer to being even. If you recall, this side was 70-30, and over here on this side it was 70-30, right? But we had 70% water, 30% sugar. Over here we had 30% water, 70% sugar. But we're a lot closer to having our sugar concentration be equal. We're also a lot closer to having our water concentration be equal. So even though we had an inability to move the sugar because of its size, water is going to be able to move in the direction from high to low concentration, move in the direction of the gradient, and we call that process osmosis. So when water crosses a membrane, the diffusion of water particles from high to low concentration across the membrane is called osmosis. Okay, so this is just to reiterate that point. Okay, so high to low concentration, and a lot of times this is going to happen when we have larger particles that can't move. Like in that previous example, we had sugar, no ability to move across the membrane, so the only way we can get closer to equilibrium is by shifting those water particles across that membrane. Okay, so now I want to just go through a couple very important terms in terms of tonicity. Tonicity is like the amount of dissolved solutes that are in something. It's a kind of a relative term. Okay, so we have 3 different solutions here. So if we take a red blood cell, a typical blood cell in a human being, we put that in straight water. Now inside this red blood cell, you got sugars, you're gonna have salts, and you're gonna have water. Most cells are gonna be upwards of 70% water, and if you put that in straight water is 100% water. So no matter what, there's gonna be more salts and sugars inside this cell than there is in this container. Okay, and that would be a hypotonic situation. If we did that same blood cell and we put it into a salt water solution, so in this case the water on the outside of the cell is saltier than the cell or has more dissolved solutes in it, salt or sugar really is irrelevant in this particular example, what will happen is the cell then would be considered to be in a hypertonic solution. If we put that red blood cell in exactly the same tonicity solution, so let's say that it's had like a 15% solute concentration within the cell, and we put it into a 15% solution, so exactly the same concentration of solutes inside and outside the cell, then we would consider that solution to be isotonic. Now if we did this with a red blood cell, let's see what the outcome of these 3 different experimental designs would be. Okay, so in the regular water solution, huge amounts of water are going to flood into the cell for the same reason we discussed in the previous slide. Okay, the solutes are not going to be able to move out of the red blood cell as you might expect, and large quantities of water are going to move in. That's going to cause this cell to blow up, rupture. In a hypertonic situation, the cell would shrivel up and, you know, become almost nothing. A lot of the water that was in that cell would be moving out of that cell to again try to reach that equilibrium. In the isotonic situation, we should see no overall impact on that red blood cell, and for that reason, like if you have a vaccination or you think about changing or cleaning out your contacts, we're going to want an isotonic solution in all of those scenarios, and therefore we should have very little change to the overall water concentration and solute concentration within cells. Let's look at that same thing for plant cells. Now, plant cells, if you recall, you have that outer cell wall, okay? So this is going to be a huge difference, okay? And so in this particular case, we put this plant cell in a hypotonic situation where there's more solutes inside the cell than the surrounding environment, and we're expecting, again, water to flood in here. But because of this cell wall, we have a much different outcome. And don't forget about this central vacuole in plants, very large, and it can hold large quantities of water. Over here we have the same scenario as we did before. It's a hypertonic situation. So in this case, we put the plant cell into salt water or something that has a lot of solutes in it. And this last one, we put the plant cell in an isotonic situation. So let's look at what happens in these scenarios. So in this case, the cell itself hasn't changed shape. But what has changed is the size of this vacuole. It's really gotten a lot bigger, and this vacuole is going to put pressure on the outer cell wall. So in terms of biology, this is called turgor pressure, the amount of pressure that that central vacuole puts on the cell wall. And this is how plants are able to maintain rigidity as they get larger, especially non-lignified plants, like plants that are, seasonal, or they don't have a lot of woody stems. This is how they can maintain their upright positioning, okay? Because every one of those cells is packed with water, and it's going to put pressure on that cell wall. Okay, over here, you notice that the cell membrane has pulled away. Central vacuole is almost non-existent there in the red. And we call this plasmolysis or plasmolysis. So the plasma membrane has broken away from the cell wall. And the cell wall, because of the decrease in pressure, is starting to collapse on itself. So if plants had this kind of situation where they didn't have enough water on the inside of their cell, what would happen is the plant tissue would start to collapse on itself. And you've all seen flowers that have wilted and have become limp. They're not able to stand upright, and that is the result of the lack of turgor pressure in the cell. Over here in an isotonic state, obviously there's no difference to the plant cell. Everything is the same as it was before, and hopefully everybody's able to pick up a couple terms here. Plasmolysis and turgor pressure, really important as we move on in biology.